mentioned, the spectrum of things that you can repair by basic excision repair is limited by the kinds of glycosylases you have. And so there's more than one glycosylases. And these are just some examples, and I don't want you to memorize the names or anything. What I want you to appreciate, though, is that there's more than one glycosylase, because there's more than one kind of base damage that basic excision repair likes to repair. There's a lot of them that are specialized for removing U. So basic excision repair really likes to get rid of uracil in DNA. This is the principal way cells get uracil out of their DNA. And there's also some other kinds of modifications that have glycosylases. So these are methylation events on A's and G's. There's a glycosylase for 8-oxo-G, another one for 8-oxo-G. So there's a number of glycosylases with different specificity, but one thing that's common among them is that they all tend to be removing bases that have kind of small modifications, right? These are not huge multi-carbon bulky adducts. These tend to be oxidative damage, methylations, and deaminations. So basic excision repair you might think of as, again, reasonably constrained in terms of the damage that it can repair, because it has to have a glycosylase that fits the damage. And it also tends to recognize small base modifications rather than large bulky atoms. Okay, so here's another structure, and what I want to show you from this structure is that there's kind of a theme in some of these recognition events. So this is the human glycosylase for 8-oxo-G, and the glycosylase is here. And then here's the DNA. And the DNA contains an 8-oxo-G base. So what are the kind of interesting features? Well, the interesting features is that it's a little bit similar to MGMT in that the damaged base, which is a little bit hard to see, but it's right here, is actually extrahelical. So like MGMT, HOG1 does this flipping of the base. So it flips the damaged base out of the helix and places it in the active site of the enzyme. And it does it in a remarkably similar way to MGMT. It binds to the minor groove, it flips the base out of the major groove side, and the enzyme also has contacts with both the damaged base, because that's what it has to repair, and the orphan cytosine that is just still sitting there in the helix once the damaged base has been flipped out. So very similar properties to MGMT. Okay, so what I'm going to do now is I'm just going to take this up a little bit of a complexity level and go through kind of the rest of the pathway. And what I want to show you is that there's actually two kinds of outcomes at the bottom of this pathway that, ultimately what you're doing is you get rid of the damaged base, then you have to get rid of the little a basic site, so you're left with a one nucleotide gap that you have to fill in with a DNA polymerase. And there's actually two different outcomes in terms of how the gap is filled. One of them is called short patch repair, which shockingly involves a very short amount of nucleotides inserted, and the other is called long patch repair, where a greater number of nucleotides are synthesized in the repair tract. So let me just kind of take you through that. Some of this is actually, all the top part is exactly the same as I showed you initially. There's a glycosylase that recognizes the damaged base and removes it. That results in an AP site. The AP site is cleaved by an AP endonuclease. In human cells, it's called APE1. The cleavage event happens on the 5' side of the A basic site. And then what has to happen, or what happens at this point, in the last thing that I showed you, because the product here is on the 3' end a hydroxyl, and then on the 5' end this deoxyribose phosphate. So the next step is that this has to be removed, and I mentioned this is done by a lyase. And so the lyase in this case is actually a domain of DNA polymerase beta. So DNA polymerase beta is another one of these specialized DNA polymerases that has a role that's very distinct from the polymerases alpha, delta, and epsilon that function in the replication fork. Pol beta is involved in DNA repair reactions like this one, and it actually has two domains. It has this lyase domain, and it has a DNA polymerase domain. So it's kind of a bifunctional enzyme. So the first step that Pol beta performs is this lyase reaction, which removes the 5' deoxyribose phosphate. Okay, so that's kind of occurring somewhere around here. So now, what you have is a substrate, again, where you have a 3' hydroxyl, now you've got a 5' phosphate, you have a 1 nucleotide gap. And so in short patch repair, it's pretty simple. All that happens is that the Pol beta that's already there inserts the 1 nucleotide. Okay, so you have just 1 nucleotide of new DNA synthesis. Now you've removed that one, or the 1 nucleotide gap has been filled in, and you're left with a 3' hydroxyl immediately adjacent to a 5' phosphate, and that's a substrate for DNA ligase. In this case, it's actually a specialized ligase that's involved in repair reactions, not the same ligase that's involved in DNA replication. Okay, so this is the short patch. Short is 1 nucleotide. The long patch is a little more complicated. The long patch seems to still, at least part of the time, involve Pol beta's lyase activity. So the steps as far as getting to this 1 nucleotide gap are the same. But then downstream of that, the filling in reaction is different. And the filling in reaction is carried out by a cast of characters that I hope should look quite familiar to you. This is basically the replication machinery. There's the clamp loader RFC, the sliding processivity clamp PCNA, and DNA polymerase delta or epsilon. So it's a little bit curious. You might wonder, why do you need a processivity factor and a very processive polymerase to insert 1 nucleotide? It's a good question. It's not really clear. What's known is that it happens. It's not really known why it happens. But when this does happen, an interesting kind of DNA synthesis occurs, which is called strand displacement synthesis. And this was what I was talking about when I mentioned earlier that Okazaki fragment maturation has some parallels in repair reactions. So what's strand displacement synthesis? It's basically this. If you have a 1 nucleotide discontinuity, the polymerase can bind here and start making DNA, and it has a couple of options. It can put in a nucleotide and just stop, or it can do strand displacement synthesis, in which the strand that's annealed to the DNA ahead of it is displaced as it synthesizes the new DNA. Okay? And this is what Pol delta or epsilon do in this long patch base excision repair reaction. So it would have started around here somewhere, and now instead of just incorporating 1 nucleotide, it's incorporated a stretch of nucleotides. But now we have a problem. We've got this, stupid 5' flap hanging off, and we need to have a nice 5' phosphate right here so that we can ligate this thing together to give a continuous DNA strand. So that property is provided by a specialized endonuclease called FEN1. And what FEN1 likes to do is it likes to cleave a 5' single-stranded flap at its base, where it makes this transition from single-stranded DNA to double-stranded DNA. So the action of FEN1 then cleaves